Well, hello everyone. Today we're going to be looking into the how genes are being expressed. So we've talked about the structure of DNA. We've talked about how this can actually be modified, things like that. So what we're going to now, that's been leading us to this particular point in the semester, and that is, how do we get the phenotype from all of these genes? How is this regulated? How is this started? How is this stopped? How is a gene turned on at certain time points? Now this is very detailed information, but we need to get you to a point to where you understand how genes are actually being expressed, how they're turned on, and how they turned off in general. So that's what we're going to be doing today. Now to do this, we actually had to get an understanding at the basic level. So after Watson and Crick did their work with DNA, and Crick continued his work with the central dogma, which is DNA, RNA, protein, then people started looking at, okay, how does this come together? And so they used a very simple organisms, bacteria, to try to address this issue about how do you get genes to be expressed in these systems. And that's where this all comes into play, this one particular chapter. What they discovered is they found that these enzymes can be regulated at the level of transcription, or proteins be regulated at the level of transcription. But remember, a lot of people back then, one of the ways to see a phenotype was to look at genes that were associated with enzymes, because then you can actually assay for the enzyme activity itself, and that's how they would do it. So we're going to be looking right off the bat. We're going to be looking at gene regulation in bacteria and how that comes to play. So, regulation of gene expression in bacteria and bacteriophages. We're not really going to go into bacteriophages, but we're looking at the bacteria. So the terms that we need to make sure you understand is, what do we mean by gene regulation? Well, that means that the level of gene expression can vary under different conditions. So depending upon what that bacterial needs at that moment in time, it's going to activate those genes. Now, this is an important concept here. You have to remember and take yourself down to the level of a bacterial cell. A bacterial cell has to be very careful about how it utilizes its genes, because they just can't keep genes on constantly, because it's a waste of energy, and they're constantly searching for energy sources to stay alive. So if they don't need a particular gene system to work in that particular environment, then they keep it turned off. All right? But if they suddenly end up in an environment where they need that gene system, they have to have a mechanism for turning that on. And that is what is evolved in bacteria to allow them to survive within their particular environments. So through evolutionary processes, organisms have developed ways to compensate for environmental changes. So after gene activity to optimize, they've altered it to grow and reproduce in a given environment. So basically, when we're going to be looking at common points of gene regulation, we're going to be looking at transcription and translation. And then there's some post-translational stuff. So this actually shows sort of a combination of all the things that come into play, not only for bacteria and prokaryotes, but also when we start looking at eukaryotes. There are some common trends in regulation. Initially here is where we're going to be focused in the bacteria. Remember, bacteria don't have a nucleus. So there are, because eukaryotes have a nucleus, there is additional levels of gene regulation that fall on top of that particular system. All right, so taking it back down to the bacteria, there are two types of genes. And those two types of genes are called, one is called a regulated gene. Now, based upon the name and the definition I gave you for regulation of gene expression, the activity is controlled in response to needs of a cell or organism. So those are going to be the ones that we're going to be focused on right here. There is also genes that are called housekeeping genes, or more appropriately, constitutive genes. They're always active, and if you think about it, it makes sense. You need protein synthesis, so you need genes necessary for protein synthesis. Therefore, you have to have the ribosomes constantly being generated. You also, for bacteria, their default mechanism for energy source is actually glucose. And because of that, they, for everything we look at, glucose, if glucose becomes available within the environment, what will happen is the bacteria cell will turn off anything else that's using a different energy source like lactose or ribose. It'll turn those off and only use the glucose. So their glucose, their genes for utilizing glucose are constantly on, and that is constitutive. All right? It's a very important point to remember as we go through here, because sometimes a bacterial cell will end up in an environment where there's very little glucose or no glucose, and instead there's a high amount of another sugar source. Like, for example, we're going to be looking at lactose, which is giving us an example of how genes are regulated, and then we'll look at arabinose later on. So these are systems that provide us with a lot of information about how genes can be regulated, turned on, turned off, and we'll find that even there's a volume control associated with it. So, basic mechanisms of gene regulation in bacteria. They have developed ways to turn off genes whose products are not needed and to turn on genes whose products are needed in each environment. That's the concept we're going to be working on today. The turning of genes on and off requires some type of interaction between the regulatory proteins and the DNA sequences. So that means whatever these proteins are that are involved in this, they're going to be coming in and they're going to be interacting with the DNA. So somehow this interaction is going to lead to either turning the genes off or turning the genes on. And it could be that there could be a negative regulation where something that represses gene expression is bound to this and turns these genes off. Well, how do you turn it on? Well, maybe there's something that will come in and affect the actual the negative regulation. Maybe bind to this protein and cause it to lose its shape and come off. If you remember, proteins interacting with DNA, this is an on and off relationship because they are doing hydrogen bonding to the actual base pairs, regions in there that fit within their particular conformation where they can bind DNA. It's called a DNA binding domain. And it's an on and off relationship constantly. You always have to remember that. It's an on and off relationship. Sometimes they have a higher affinity, they stay on longer, but they'll pop off. Or they have a low affinity, they stay on shorter. Then they pop off and they're constantly going off and on. Okay? So keep that in mind as we study this. So, an overview of transcription regulation. The most common way to regulate gene expression in bacteria is by influencing the initiation of transcription. The rate of RNA synthesis can be increased or decreased. Transcriptional regulation involves the action of two main types of regulatory proteins. So this is where we're going in. So as I mentioned, they can be repressed. Now we're going to look at the first model we're going to be looking at, which was the first one identified, is going to be dealing with repression of gene expression. So this binds to the DNA and inhibits the transcription of that particular gene that's associated with that. But we also have activators. They bind to the DNA and increase transcription, and in the end we'll look at that particular one as well. So negative control refers to transcription regulation by a repressor protein. Positive control refers to regulation by an activator protein, something that binds to it to turn the gene on. Okay? So we'll learn about both of those as we go on. Okay, so let's look at this regulation of gene expression. Well, if you're going to be turning a gene on or turning it off, then this gene has another term for it. You're going to be calling it an induction. You're going to induce that gene to have an expression of its product, its protein product. And so these are called inducible gene expressions. Now, based upon the model that we're about to study, what we have discovered is there are basic structures to the gene associated with these bacteria that you need to understand. Well, you know about the terminator already. We've talked about that before. And you know about the coding sequence. That's the part that's going to give rise to the polypeptide. And you know about the promoter. And that's in front, that identifies where the start site of transcription is supposed to be. Now the one that we're going to be introducing into here is something that's called a controlling site. It's basically something that sits on top of the transitional point between the promoter and the start site of transcription. It is here where we have induction that can occur, okay? So somehow this system here, depending on where it's located, if we induce gene expression, then we're going to get a protein produced, okay? So when a gene is turned on by the additions of a substance, it is called an inducible gene. What that means is something has been added into the environment that this gene is going to respond to. Now the regulator substance is called an inducer. So the substance is an inducer that is now present in the environment, which is a member of a class of small molecules. Usually the inducer is a small molecule. We'll find that the one that works in the first gene that we look at is called allolactose, which is a conformational modified form of lactose. And that is a very small molecule. We refer to it as effector. It's a class of small molecules called effectors. And in this particular case, it is an inducer. It induces gene expression. The controlling site is near the protein coding sequence, so that's that. And the addition of the inducer leads to the induction of gene expression. So when that inducer or that effector, which we'll be talking about as allolactose in our particular first case, that effector comes in and it acts as an inducer and it induces gene expression. All right, so an effector molecule, small effector molecule may inhibit transcription too. So co-repressors bind to repressors and cause them to bind to DNA. Inhibitors bind to activators and prevent them from binding to DNA. So we're going to see effector molecules can act as inducers or they can act as inhibitors in one way or the other. Genes that are regulated in this manner are called repressible genes. So we have inducible genes which can be induced to be activated, but we also have repressible genes that can be with the effector molecule, can inhibit the expression of the gene, and that's called a repressible type of gene expression. So, binding sites on genetic regulatory proteins. So here you have a promoter, here's a repressor protein. So this is repressing gene expression, it binds here. Well, along comes an inducer, and it binds to this and causes it to lose its ability to bind to that controlling site that's sitting in top of the promoter. And as a result, it comes off. Well, while it was there, it actually blocked the RNA polymerase from even being able to bind to the promoter. Thus, when it comes off, now the RNA polymerase can come in and we can have an induction of gene expression. So, in the absence of the inducer, the repressible protein blocks transcription. The presence of the inducer causes a conformational change of the protein, which eliminates its ability to bind to DNA, that inhibits the ability of the repressor protein to bind to DNA, and transcription proceeds. So that is the model we're about to go into. Okay? All right. Now, an activator protein cannot bind to the DNA unless an inducer is present. Now, this is going to be the second model we're going to be looking at, and the third model we're going to be looking at. In this particular case, primarily the third model. In this particular case, you have a protein that's here, and that particular protein will not bind to this and bring about a gene expression. However, if the inducer comes in and binds to this, it changes the shape of this, binds to this, and that promotes the RNA polymerase coming into the correct location. So we'll look at that particular one in the second and third model. Okay? So the activator protein cannot bind to DNA unless an inducer is present. When the inducer is bound to the activated protein, that enables that activator protein to bind to DNA and activate transcription. So now you've been introduced to an inducible gene expression, you've been introduced to a repressible gene expression. So an activator protein, inducer molecule, inducible gene comes into play. All right. So the binding site of genetic regulatory protein. In the absence of a co-repressor, and this happens too, will not bind to the DNA. Sometimes you have to have an additional protein help out to bind to these. When the co-repressor is bound to the repressor protein, a conformational change that occurs allows the repressor to bind the DNA. So this co-repressor can come in later and help with this and block that actual gene expression. All right, so the activator protein will bind to DNA without the aid of an effector molecule. Sometimes this happens. The presence of the inhibitor causes a conformational change. So sometimes it's activated, doesn't need, and it doesn't have any type of effector molecule. And we'll see this later on as well. It binds and it allows for the gene expression. The presence of the inhibitor comes in, binds to it, and causes it to come off. Okay? Now, the second particular model that we're going to look at, it's called the tryptophan operon. In that particular one, it's actually a feedback mechanism that comes back in the production of tryptophan, because the tryptophan operon is all about producing the amino acids, in this case tryptophan. And if there's too much tryptophan, it comes back, binds to this, and pretty much represses gene expression. So this is where this comes into play. Okay? So this inhibits transcription. All right. So this is what we've been leading to. We talked about the variations of this whole thing that we can see in the regulation of gene expression. What we've been leading to is the original model for gene expression that was provided in the early 60s by a particular two scientists, Chpt and Rohde, and they eventually won the Nobel Prize for this. So it's called the operon theory, and I'll explain what an operon here is in just a moment. What we're about to study is the bacterial models of gene expression. To aid you in your understanding my lecture, I am providing a link. So once I go through the lecture, you can come back here, or if you want to do it now, there's actually a really good article, and if you click here, you can actually go there, and it's an article based upon what we're about to read about in the lac operon, and it goes into some really good detail about how this whole thing works. Okay, now you could watch it ahead of time, and it's got two animations on it, by the way. There are two animations that will give you some information. One animation is called the lac operon. The other animation is going to be the next model we're going to look at, called the tryptophan operon. And so if you watch that first, then as I go through this might give you a little more detail, especially because of the moving part of this, the animation part of it. It'll help you understand what I'm talking about. So I'm going to stop for a moment in case you want to go ahead and pause the video so that you can go to this particular link and look at this particular system. So I'm going to stop and then come back, and then I'll flip the slide over. Okay, well, let's get into that system. So what we're going to be talking about is the utilization of lactose by E. coli in the environment. So here's the setup here. As I mentioned before, the default mechanism for gathering energy from the environment is through glucose. Bacteria cells, all of them do this. They prefer glucose. It's the simplest structure of an energy molecule that they can bring in and utilize very quickly. So they have those genes all are constitutively being expressed. Okay? Well, what happens if a bacterial cell swims into an environment where there is no glucose? Okay, well, it needs energy, and so it has to make an adjustment, and that's where this comes in. What if it comes in a very common sugar molecule, which is a disaccharide? It's called lactose. Now that lactose, if it can bring that lactose into the system, that will activate certain genes that will now break that lactose up into monomers, which then can go into glycolysis as well as through the Krebs cycle. But it has to have the enzymes necessary to break up that disaccharide. Okay? So that's the concept where we're working at. We have moved out of the glucose environment and we have moved into a lactose environment. Okay, so that's where we're sitting right now. Okay? Now, E. coli, as you know, can grow in simple media containing salts, nitrogen sources, and glucose. That's all they need, and they can grow in that. Okay? Now, as I said, if lactose suddenly is added instead of glucose to that environment, so there's no glucose in case, for example, we don't put it in the media, we just put lactose, a number of enzymes have to be rapidly synthesized. Now, here's the interesting part about these particular enzymes that can utilize lactose. A small amount of them are always present in the bacteria, a very small amount. So that means that built into this bacterial cell, there are going to be 3 enzymes, in this case for the utilization of lactose. So one of them is actually going to be in the actual bacterial cell wall. It's called permease, and we're going to see that's associated with this operon system. Another one is going to be beta-galactosidase. So this permease allows lactose to be pumped into the cell. Beta-galactosidase is the enzyme that is going to break down that disaccharide into 2 molecules that can enter into glycolysis as well as the Krebs cycle. So it breaks it down, the lactose, into galactose and glucose. Now, this is not always a very precise mechanism of breaking down this lactose. Sometimes it will bind to it, quickly bind to it, and instead of breaking the bond, the covalent bond between the 2 molecules of sugar, what it does is it twists. It conformationally modifies that bond so that it ends up having a stereoisomer that is a little bit different than the original one, as you can see here. As a result of that, this creates a stereoisomer of lactose called allolactose. That's very important because if that allolactose is not bound to the beta-galactosidase again to break it up, then what happens is if there's enough of them, they will go to where the genes, where these particular genes for beta-galactosidase and permease, and there's another one, transacetylase, are located, and induce more beta-galactosidase and permease to be produced by the cell. To overcome this problem that it has, it doesn't have enough of these enzymes necessary to break down the lactose. So that's the concept that we're about to move into. So when lactose is the only sugar in the environment, 3 enzymes are synthesized: beta-galactosidase, lactose permease, transacetylase, which is acetylates the beta-galactosidase, and it functions in the lac arm is not completely understood, but these 3 come about. Okay, now they're already there, they're already in the system, but at a very, very low amount. Their job is to be in that low concentration. Their job is to respond to the environment if there's a change. If there's suddenly no glucose and it's all lactose, then what they do, they're fired up and they start to activate their own gene expression by making these modifications of the lactose. So what happens is lactose is converted into another disaccharide called allolactose. And guess what? Allolactose is going to be a class of part of the effector molecule class, and it's going to be the inducer. Allolactose is an inducer, not lactose. Allolactose is the inducer. Very important that you remember that. Okay, we're going to take a break in this particular part of the video, and then we're going to come back and we're going to start the system up.